So, good morning, everyone, and welcome to the EvoFunPath webinars. So today we have a distinguished guest, as she's one of the renowned fungal geneticists, in addition to being an infectious disease clinician. So it's my pleasure to introduce you Dr. Julia Köhler, who's an associate professor of pediatrics at Harvard Medical School. So Julia, correct me if I'm wrong, but Julia received her medical doctorate from University of Heidelberg in Germany, and then trained in pediatrics and pediatric infectious disease at Boston Children's Hospital. She studied yeast genetics and Candida molecular biology in the lab of Gerald Fink at MIT, and then started her own lab at Boston Children's Hospital. So for those who work on Candida and yeast, Julia and her colleagues invented the spider medium that many of us are using routinely to promote yeast fermentation or biofilm formations. Julia labs study nutritional signaling pathways in the human fungal pathogen Candida albicans and their contribution to virulence. Julia also made many useful plasmids that are widely used among members of the fungal community. On the clinical side, Julia is particularly interested in pediatric infections. Her work also encompasses a focus on immigrant health and social justice, particularly the protection of immigrant rights. So before leaving you the stage, Julia, I just wanted to notice our trainees that they can ask questions directly at the end of the seminar or send them to me in the chat box and I will take care of communicating them. So Julia, you are very welcome, and we look forward to hearing your talks on fungal pathogen response to phosphate starvation.
Thank you so very much, Adnan, for your too kind introduction. I am excited to be at EvoFunPath, which I now know is an amazing program to collaborate, inspire each other, meet each other in person, and train the next generation of fungal biologists. So it's just a big honor to be here with you, and I appreciate your presence. So as Adnan said, I study Candida albicans, but what I'm going to be talking about is actually relevant for a lot of other fungi. And given what I just learned about EvoFunPath and its members, I'm just going to sneak in a few mentions of that as well. So first, I want to thank the amazing people who did the work that I'm going to show you. Tamina Chowdhury was a fantastic postdoc who started the Target of Rapamycin work that I'm briefly going to touch on. Ningning Liu did a lot of the Pho84 phosphate transporter work, which was then continued by Catherine Chi and Michael Acosta Zaldivar. And contributions were also made by Niketa Jani and Udita Roy, also with the help of fantastic collaborators at Children's Hospital. And I just want to highlight the brilliance and hard work of each one of these wonderful people. Similarly, a lot of collaborators in many different institutions brought their own expertise and thoughts to bear. And in the interest of time, I'm not going to mention everyone, but I definitely want to acknowledge that without them, we would not have learned what I'm going to be telling you about. So as Adnan mentioned, Candida albicans is a fungal pathogen, an opportunistic pathogen. I think as EvoFunPath members, you've heard a lot about it, so I won't dwell on it. Just to mention, it does have this capacity to switch frequently between growth as budding yeast and growth as filamentous hyphae. And one starting point of our work was when we realized that Target of Rapamycin regulates these morphogenetic switches among other regulatory systems. And that led us down the path that I'm going to tell you about. The topics I'm going to touch on is how we discovered the major Candida phosphate transporter, Pho84, how we realized that this transporter, Pho84, is required for Candida cells to withstand oxidative and cell wall stress, and then, thinking about could there be a mechanistic connection between the cell's ability to withstand these stresses. The question then came up, Pho84, based on just genome scoping, has 4 inorganic phosphate transporters. Why does deletion of one have these significant phenotypes? And lastly, I'm going to leave you with an idea of in vitro evolution as a forward genetic screen. So that's what I'm going to be touching on today. Oops, okay. And this is kind of a summary slide just to let you know the concept. So Pho84 is the phosphate transporter that is going to be at the center of the talk. And we found that it signals to a small GTPase that activates the important regulator Target of Rapamycin complex 1, which resides on the vacuole. And a superoxide dismutase that is important to dismute superoxide into H2O2 and water is actually regulated by TORC1 is what we found. And that was one mechanism we discovered how Pho84 contributes to oxidative stress resistance of Candida albicans. With respect to the cell wall, there are cell wall polymer biosynthetic enzymes that reside inside the Candida plasma membrane. They use nucleotide sugars as their substrates, and these nucleotide sugars come from central carbon metabolism and require phosphate because they are nucleotides, and that is a major role of Pho84 in the cell wall stress resistance system, because if these substrates are not available, the cell wall polymers will not be made. So why phosphate? Well, I just downloaded this schematic of a eukaryotic cell from a random website. And when you look at every single organelle, I'm just going to go around, you realize that it requires phosphate. So in the mitochondrion, I keep searching for the highlighter. Sorry about that. Okay, I'm going to leave the highlighter and just say in the mitochondrion, we have phosphate pumps that provide the inorganic phosphate for ATP synthase. ATP has 3 phosphorus atoms and 3 phosphate residues. If the ATP synthase does not have that inorganic phosphate available, ATP will not be made and the mitochondrion will not do its work. The ribosome consists to a large degree of RNA and ribosomal RNA. And what is the backbone? How are these nucleotides held together? By phosphate moieties. The ER, the Golgi, and the plasma membrane consist of phospholipids. How do phospholipids connect their nonpolar and their polar moieties? Through phosphate. The nucleus and its content, which are chromosomes, like RNA, DNA is basically held together by phosphate moieties. The nucleolus, which produces the ribosomes, again, uses RNA and therefore phosphate to make this ribosomal, to produce these organelles. And I'm just going to mention that the vacuole is actually a storage space for phosphate in fungi and potentially also in humans, but we're going to be talking about fungi. And so the other important part, let's just say here it just says cytosol. Well, what happens in the cytosol? Poor metabolism to provide the intermediates that all these biosynthetic processes must use requires phosphate at so many steps. So even just to keep glucose in the cell and to start glycolysis, the pentose phosphate pathway, the biosynthesis of polysaccharides, you must phosphorylate the glucose that you have imported to glucose 6-phosphate. And when if you want to go through glycolysis, you must add a second phosphate. So this is from an interesting review by Sunil Laxman, who postulates that metabolism is a source and a sink of phosphates depending upon the availability of phosphate to the fungal cell. So everywhere where there's a green, phosphate can be released. Everywhere where there's a red color, it must be, it is a sink. I'm not going to go into all of that, just to say for every part of the processes in this living cell, phosphate is actually required. Really, it's hard to think of anything where it's not. So quick circle back to Target of Rapamycin. Not everybody thinks about it or is familiar with it. It is actually the brain of the cell, as its discoverer Mike Hall called it. And what it does is I know Adnan thinks about it a lot, but not everybody does. So just quick mention, when the cell should be happy because it has plenty of nutrients and it is not under stress, TORC1 collects that information, Target of Rapamycin complex 1, it collects that information and activates growth and proliferation processes. When the cell should be unhappy because there's stress of any kind, or when a bacterial product, rapamycin, binds to TORC1, then stress responses are activated. These processes are stopped in a regulated manner. For Candida, hyphal morphogenesis is induced. So this is a very global regulator of multiple processes in the cell. And because it keeps close track of the nutritional status, we ran into it in a mutant screen. What we did for this mutant screen was we took our NAT1 dominant selectable marker and we marked a Mariner transposon with it. We in vitro transposed using transposase the transposon into a Candida genomic library such that there were now a bunch of differently at random insertion sites marked genomic fragments, and then we transformed that into Candida albicans. This works because Candida is known to be haploinsufficient for many genes. In other words, if you have a transposon disruption of many genes, you will detect a phenotype. So we use the Mariner system because it is maximally random and more than other transposons. It's been used a lot in bacteria. And we screened this collection of mutants for rapamycin hypersensitivity because we wanted to find more TORC1 connected processes. And we identified a transposon insertion in the phosphate putative at that time phosphate transporter Pho84. It was just a like a homology annotated gene in the genome database. And when we made targeted deletion mutants in this gene, in Pho84, in fact, they were also rapamycin hypersensitive. The heterozygote also has a haploinsufficiency defect, as does the reintegrant. So the targeted mutant did recapitulate the transposon mutant phenotype. And when we then tested their behavior on phosphate starvation medium, these Pho84 null mutant strains had a severe growth defect, suggesting that Pho84 truly is important for growth of Candida albicans in phosphate scarcity conditions. And because we discovered it in connection with TORC1, we wanted to see whether it is important for TORC1 signaling. Our readout for TORC1's activity is the phosphorylation state of ribosomal protein S6. And so we tested protein extracts from wild-type cells, Pho84 deletion cells, and reintegrants for TORC1 activity or phospho-S6 levels in this Western blot at different phosphate concentrations. And we saw that, in fact, at every concentration, TORC1 activity is lower in the Pho84 null mutant, although it does rise with more availability of phosphate if you compare this to this band, but it is always lower than the wild type. So we concluded that Pho84 is in fact required for normal TORC1 activity. We wanted to see whether the opposite is also true, because TORC1 signaling often regulates nutritional processes. So we made mutants in which the TOR1 gene, the target of rapamycin kinase encoding gene, is controlled by TERO, a repressible element that responds to doxycycline or tetracycline concentrations in the medium in a titratable way. And what we saw was that repression, and then we also made a truncation mutant where the first protein-protein interaction domains of the TOR1 kinase are deleted because those are the least conserved domains between human and the Candida kinase. What we saw was in these mutants, Fo84 expression was abnormally increased when we repressed TOR1 expression. In other words, in rich medium, TOR1 is required to repress Fo84 transcription, indicating a connection between the so-called Fo-regulon that regulates acquisition of phosphate and TOR1. Now, just briefly, we were interested, it's a pathogen. We always want to know how does a gene impact pathogenesis? Long story short, in a Drosophila model, we saw that the 44 null mutant was significantly less virulent than the mutant or the reintegrant. This was work in Per Ljungdahl's lab in Stockholm. And then in Scott Filler and his team also examined behavior of these mutants in a oropharyngeal candidiasis mouse model and saw that the mutants and the reintegrant had a decreased fungal burden in the tongue tissue that was infected with these different genotypes. So this was interesting because, it's a phosphate transporter. What does that have to do with virulence? So we looked at various stressors that Candida might experience in the host. Among them, cell wall stress, heat stress, and oxidative stress. And what we found was that actually, in fact, cells lacking Fo84 are hypersensitive to cell wall stress imposed by the echinocandin micafungin. They are hypersensitive to the chitin binding compound Congo red. They're hypersensitive to heat stress. And suggesting that there are stressors that, and the heat stress can be rescued by sorbitol, suggesting it is not an osmolarity issue. So collaborators in Neil Gau's lab measured the cell wall thickness of these mutants versus the wild type, and they do these amazing electron microscopy sections, and they actually measure for different cells these layers, the outer and the inner layer, and the null mutant had a significantly thinner outer layer and a statistically non-significant thinner inner layer as well of the cell wall. So the cell wall is thinner in the Fo84 null mutant. And when we measured ourselves various components of the Candida cell wall, which are polysaccharides, several of them were decreased in the Fo84 null mutant, including chitin and beta-1,6-glucan and phosphomannan. We didn't have the ability to measure beta-1,3-glucan, but I assume that it is also decreased. So thinking about how this could be, we performed metabolomics experiments where we starved wild type and Candida cells for phosphate and then refed them with low or high phosphate concentrations in the media and looked at what kind of metabolites are measurable. This is a follow-up experiment showing that where we just simply grew cells in ATP and then in a different assay, just measured in a direct assay, measured ATP, and we found that the Fo84 null cells are depleted for ATP compared with wild type, and the reintegrant is still depleted, presumably because it's haploinsufficient. And what we also found, and what was pretty striking, was that for sugar nucleotides, where two of them, UDP N-acetylglucosamine and UDP glucose, are substrates for our cell wall biosynthetic enzymes, the intermediates that come after phosphate addition steps, where another phosphate is added, were depleted in the mutant compared to the wild type, whereas the precursors before phosphorylation were increased. UDP N-acetylglucosamine is the substrate for chitin synthase. UDP glucose is the substrate for beta-1,3 glucan and beta-1,6 glucan synthase. So one simple idea was that these enzymes do not have their sufficient substrate, and so they are not able to make the cell wall components that lend the cell wall structural stability, and therefore these Fo84 null mutant cells are hypersensitive to cell wall stress. So this is depicted in that schematic. Here are the precursors. Here are the biosynthetic enzymes. They just don't have their substrates, and so the cell wall is going to be thinner and more susceptible to stress. The other stressor that we looked at was oxidative stress, and for both for superoxide ion generators and for peroxide, the null mutant was hypersensitive to these oxidative stressors. And when we measured reactive oxygen species of these cells without adding any external oxidative stressors, we found that they were also elevated at every external phosphate concentration. So reactive oxygen species are increased in Fo84 null cells. We were wondering whether maybe the oxidative stress signaling system of Candida, which is largely provided, or a major component of which is the HOG1 system, might be defective, but what we actually found was that the opposite is true. When cells are exposed to H2O2, the response of Fo84 cells is stronger and persists longer than that of wild type cells. So it's not that they cannot detect and respond to oxidative stress, they just have more oxidative, basically reactive oxygen species in their cells. And we tried to figure out, how can that be? One kind of obvious path of thought was to look at the TOR pathway, because that's how we found Fo84 in the first place. So there were hints that a GTR1, a small GTPase that activates TORC1, could interact with Fo84. So we simply overexpress GTR1 and also a GTR1 active locked GTP rather than GDP locked mutant in wild type and Fo84 null mutant cells. And what we saw was that, surprisingly, these cells that are kind of have hyperactive TORC1 have decreased reactive oxygen species even in the 484 null cells. So this is wild type, this is the 484 null. And they also protect cells from increased reactive oxygen species during rapamycin exposure. So one idea was that actually it is the 484 null cells decreased torque signaling that and disables their ability to handle these reactive oxygen species. Now, a important system for dealing with superoxide anions that I was mentioning briefly before are the superoxide dismutases in the Candida cell and also on its surface. We looked at the internal, like the cytoplasmic and mitochondrial superoxide dismutases, and one that was clearly diminished in 484 null cells was SOD3. So SOD3 is a cytoplasmic fungal, kind of fungal specific superoxide dismutase, and in 484 null cells, just the amount of it was substantially decreased. You know, since overexpression of GTR1 had this effect on reactive oxygen species, we wondered whether it could rescue this defect of SOD3 availability. So we used the GTR1 overexpressing cells to measure SOD3, and in fact, 484 null cells that overexpress GTR1 did have substantially higher SOD3 levels than those that contained the cognate vector, indicating a mechanism by which lack of 484 might contribute to the cell's inability to handle oxidative stress. And that's depicted in this cartoon. So 484 signals to GTR1. GTR1 activates TORC1, and then in some way that we don't yet know, TORC1 induces SOD3 expression. Now, superoxide comes from the outside. It arises during metabolism in the cell, but it is importantly produced by our own immune cells, the most important ones against Candida being the neutrophils, by a dedicated enzyme that releases superoxide onto the Candida cells when neutrophils engage with their target pathogen. So one idea is that TORC1 activity is required for SOD3 to cope with the superoxide anions that the neutrophil is bombarding the Candida cell with. So however, we have this pathway, 484, GTR1, TORC1, SOD3, but that is not the only relevant pathway here. So we looked at whether overexpressing GTR1 rescues hypersensitivity of 484 null mutant cells to killing by whole blood and by neutrophils. And long story short, the answer is no. So here are the cells overexpressing GTR1. And here this is, I guess, the better, clearer phenotype. We isolated neutrophils from human volunteers. So the 484 null cells containing the vector are still killed by the neutrophils, as are the 484 null cells overexpressing GTR1. So overexpressing GTR1 does not rescue their hypersensitivity to neutrophil killing. And similarly, overexpressing SOD3 also does not rescue killing by whole blood of Fo84 null cells. They're still hypersensitive to it. So although, of course, these immune cells have other mechanisms other than oxidative stress to kill the pathogens they encounter, we still thought there's got to be something else going on. And, I've been pondering this for a while. So looking back at the metabolomics results, one thing that is striking is that many of the metabolic pathways impacted by lack of Fo84 are central carbon metabolism pathways. And I'm just going to highlight glycolysis here, the TCA cycle, and importantly, somewhere is the pentose phosphate pathway. Pentose. Of course, these pyrimidine and sorry, this is pyruvate, but pyrimidine and purine metabolism is what generates sugar nucleotides, but other component like the substrates of our cell wall biosynthetic enzymes, but other components of the core carbon metabolism are affected by lack of Fo84. So that got me thinking again about the importance of phosphate for carbon metabolism. And this cartoon simply depicts the pentose phosphate pathway and glycolysis and how they interact. One molecule that I was thinking about with respect to oxidative stress is NADPH. So NADPH is produced to the largest, let's say, fraction of the available NADPH in the cell in the oxidative part of the pentose phosphate pathway in the first step and in the third step here. Why is that important? And one more thing I'm going to mention is that ribose 5-phosphate is also produced in the pentose phosphate pathway, and that actually is a metabolic intermediate for the biosynthesis of NADP. So NADPH is an important okay. I'm going to get to that in a moment. But when I look back at the measurements of NADP in 44 null mutant cells and compared to wild type cells, there is really a stark difference. It's like a roughly threefold difference that is highly significant. Now, these are the metabolomic measurements. So I've just been thinking about this recently, so we haven't measured directly NADP or NADPH concentrations in these mutant versus wild type cells, but thinking about NADPH, so it has these three phosphorus atoms here, 3 phosphate moieties, and it has the ribose, which it gets from the pentose phosphate pathway. And the reduction of NADP to NADPH occurs in the pentose phosphate pathway, which together with glycolysis is deranged in Fo84 null mutants. So one consideration that we haven't looked at is, does lack of NADPH in the Fo84 null cells further disable their ability to deal with oxidative stress. We looked at metabolism in the context of cell wall stress, but could it actually be impacting oxidative stress and their inability to synthesize sufficient NADPH renders them hypersensitive to oxidative stress? How does that work? Sorry, I think I'm missing a slide, and I'm just going to say what we need NADPH for, and that is to provide the cofactor for thioredoxin reductase. When these SOD when these superoxide anions encounter a protein or another large molecule, they will oxidize the protein's thiol residues. In other words, if you have a cysteine with an H on it and another cysteine in that same protein, that SOD will oxidize them. They will form a sulfide bond, and the whole protein is going to be distorted in its tertiary structure massively and disabled. So that's the job of thioredoxin. It uses its own reducing equivalents to, again, reduce those two cysteines, open up the protein to its original structure, and then the cell doesn't want to discard that precious thioredoxin, so it reduces it with an enzyme called thioredoxin reductase, and that uses NADPH as its cofactor. So oxidative stress actually induces the pentose phosphate pathway. It activates the first enzyme in that pathway in order to produce NADPH as reducing equivalents for the cell. So that's my newest line of thinking about how FO84 might impact oxidative stress management in Candida, and we have to kind of delve into that a little more. At this point, it's just an idea. And what we do see is that the levels of this intermediate or of this cofactor are dramatically reduced in the FO84 null mutant. So switching gears for just a moment to wrap up, genome scoping shows 4 phosphate transporters in the Candida genome. How can it be that FO84 has all these phenotypes when there are 3 other phosphate transporters, inorganic phosphate transporters? In work done by Michael Acosta Zaldivar, and together with Catherine Chi, we deleted each of these transporters, but then we also made mutants in which only one of them was left behind and 3 of them were deleted. So these are triple mutants. Here, the mutant has FO84 alone, and these 3 transporters are deleted. Here it has FO89 alone, etc., etc. So this A stands for alone, and we use these alone triple mutants to characterize their activity at different pH levels. Why look at pH? Because it's known from Saccharomyces that pH strongly influences the behavior of phosphate transporters. The reason is that FO84 is a proton symporter. In other words, to drag in a phosphate, it needs to drag in a proton as well. Whereas FO89 uses sodium symport. Long story short, in the interest of time, what we found is that at all these different pH conditions, the one that enabled growth the most was Pho84. This is at high phosphate, this is at low phosphate. Each histogram bar represents the area under the growth curve of 3 biological replicates for these alone mutants under these pH conditions. So in terms of enabling growth, it is really Pho84 that looks the most like wild type and that most likely represents the majority of the growth enabling phosphate transport. And at high pH, like 6 or, getting to neutral, 6 or above, Pho89 also contributes significantly, not more than Pho84, but it does contribute, whereas these others really are active only at high phosphate level. Pho87 and FGR2, they are low affinity transporters. So Pho84 contributes both at high and at low phosphate concentrations, more than any other phosphate transporter, to the ability of cells to grow. And then Michael measured directly phosphate uptake by measuring the phosphate concentration of the medium over time. And basically, the Pho84 alone cells resembled the wild type most closely at these different pHs, whereas the other transporters really depleted the medium more slowly, and Pho89 was active only at alkaline pH, whereas at acidic pH, it really was not able to remove phosphate from the medium. We wanted to see whether Candida can adapt to lack of phosphate over the long term. This is a 2-month experiment. We passaged populations every 2 days. We had 2 transformant isolates of quadruple mutants, where we deleted all 4 of the inorganic phosphate transporters, and we passaged them over time. This is area under the curve, under the growth curve. These are the growth curves, and they are represented by these histograms. And we saw that 2 different lineages were actually able to some extent, not fully, regain fitness in low phosphate conditions. So this is 0.4 millimolar, not, extremely low, but also kind of scarce for the cell. And in terms of their ability to regain resistance to the stressors we were interested in, one of the lineages regained cell wall stress, let's say, resistance, kind of to the same extent as Pho84 null cells, whereas the other did not. And then similarly, the same thing for oxidative stress. And for rapamycin, interestingly, with respect to TORC1 signaling, target of rapamycin signaling, there were disparate phenotypes where the more stress-sensitive lineage became more tolerant of rapamycin. And this was due first to large scale and then to large-scale genomic rearrangements, which in the interest of time, I'm just gonna skip over, and then to small-scale mutations that affected, interestingly, target of rapamycin 1 kinase, a kinase that is thought to signal to TORC1 called PCK2 and some others. So to wrap up, our thinking right now is that there are links from FO84 to TORC1 signaling that enable SOD3 expression to detoxify superoxide anion. Phosphorus is simply needed as a component of these intermediates for cell wall biosynthesis. And there is also a dinucleotide that contains 3 phosphate residues, and that is critically important for the cell to remove oxidized to restore thiol residues to oxidized proteins. I'm going to say it that way. And this mechanism that connects metabolism with oxidative stress resistance is something that we need to explore further. And with that, I will end and thank you for your presence.
Thanks, Julia. Incredible story and tremendous amount of work as well. Is there any question for Julia? Well, let me start with the first, probably. So yeah, Julia, so if you mentioned that there's a connection between, so the connection between the TOR pathway and the SOD3, like the superoxide dismutase. So is there any idea how this is connected?
Well, I think that the physiology behind it is that TORC1 activates anabolic processes. In other words, everything that contributes to growth and proliferation of the cell, TORC1 favors it. TORC1 activity favors it. Now, when you have electron transport chain activation and oxidative phosphorylation, you are going to generate reactive oxygen species, superoxide, because some of that oxygen leaks out from where it's supposed to be in the mitochondrion, and will start producing superoxide and reactive oxygen species, so that when you have active metabolism, you are going to have reactive oxygen species in the cell. And maybe as TORC1 is activating metabolism in kind of an anticipatory way for this oxidative stress that the metabolism itself generates, it's going to induce superoxide dismutase expression to kind of be ready as a sponge for those reactive oxygen species. So that's my speculation.
Okay, thank you. Any question? Yes, Isabel?
Hello, thank you very much for this very interesting talk. I'm Isabelle Benoit from Concordia University in Montreal. And I was wondering if maybe I missed it, maybe you mentioned it, but is the transporter conserved throughout the fungal kingdom?
That is a very, very interesting question. Thank you for that, Isabel. The answer is yes. So the most detailed structure for FO84 homologs comes actually from a mycorrhizal fungus, whose name I'm going to mispronounce, Pirisporiforma something. And it is conserved. It is, and when you just line up the primary amino acid sequence of these different fungal species, Pho84 homologs, they are highly conserved. Now, why is this? Because fungi are osmotrophs. They must take up their nutrients individually as small molecules. They can't just engulf something that they're then going to digest internally, like, let's say, an amoeba does or a human does. When an amoeba engulfs a bacterium, it's going to get phosphate, it's going to get, nitrogen, it's going to get carbon, but a fungus does not. So in the environment, phosphate, although it's quite abundant, it is insoluble. It is bound to organic and inorganic other molecules. For example, phytic acid is one of the organic molecules that contains a lot of phosphate, but is not accessible to an environmental fungus like these mycorrhiza. And that's why every fungus that I've ever looked at has a closely related Pho84 homolog, because they all have to take up soluble phosphate up a steep concentration gradient in order to keep these processes going. And so, as we know, the mycorrhiza are cool fungi because they expand the plant's access to critical nutrients like nitrogen that the plant doesn't easily get enough of, and importantly, inorganic soluble phosphate for the plant, and the mycorrhizal fungus enables the plant to acquire the phosphate that it needs.
Thank you very much. Is that true for the other one as well? The other transporter?
That I don't know.
The
That's
one you
a great
mentioned that were less
question.
effective.
Yes.
You mentioned yeah.
Yes, my suspicion is no, because even between Saccharomyces and Candida, the low affinity transporters are a little different, but I have not looked at that.
Thank you very much.
Thank you, Isabel.
Hey, Julia, just a question connected to what Isabel was asking about. So for 84 and at a certain level 89 are somehow connected to phosphate uptake. What about the others? Any idea what would be their function? Or
Yeah, so the low affinity phosphate transporters are that's also their job. They take up phosphate when there's abundant phosphate, soluble inorganic phosphate in the environment. And in Saccharomyces, they're actually degraded when phosphate becomes scarce, because they might let it leak out, actually. So they might let it go out in the other direction.
yeah.
So they are there when phosphate is abundant to allow the cell to acquire a lot of phosphate, because the cell has various ways to store phosphate that it's not yet using. So when the phosphate is abundant, the cell loves to take it up and to just store it.
Okay, thank you. Any other question? Come on, guys. Don't be afraid. Okay.
I'm just going to say that, osmotrophic organisms, including bacteria and plants, have similar phosphate homeostatic systems as fungi do. And in plants, it's definitely linked to TORC1. Bacteria don't have TORC1, it's a eukaryotic system. And again, that's because they have to take up each nutrient individually, so phosphate is a big challenge for them. because its concentration, the soluble phosphates concentration in the environment is somewhere between 1/10 and 100th of that in the cell.
So maybe one last question. So then I will leave you with our trainees for more casual discussion, I guess, for a couple of minutes. So just one last question, Julia. So phosphate uptake, is it subject to transcriptional regulation? In other words, is there any transcription factor that govern, phosphate uptake?
Yes, absolutely. There is a very tightly regulated transcriptional system. In Candida, the transcriptional regulator that induces genes required for phosphate uptake during phosphate starvation is FO4. It induces phosphate transporters. It induces importantly, FO84. So FO84 is transcription is very tightly regulated by phosphate availability around the cell. When phosphate is high, FO84 is downregulated and like transcriptionally downregulated, and also at the protein level. In other words, at the expression at the cell surface. And also phosphatases, because the cell tries to release phosphate from its organic molecules with these phosphatases that it secretes into the periplasm. And there are numerous other co-regulated processes that are controlled by FO4. In Saccharomyces, there's actually another co-regulator, FO2, that is required for activity of FO4. And the FO4 activity is in Candida and Saccharomyces controlled by its localization, which is controlled by phosphorylation through a CDK system, the FO85, FO80 system, and that is regulated by the phosphate availability to the cell. How that happens, how this CDK system is regulated is still, being worked out, but we know that inositol pyrophosphates are involved in that.
Okay. Yeah, thanks, Julia. Thank you again for accepting the invitation. I'll leave you, I think, on behalf of the PI and the trainees of the EvoFunPath. Thank you so much. Merci beaucoup. And I'll leave you with our trainees, I think, for more, as I see, casual discussion. So Aileen, are you here? Yes, you're here. So I'll leave you I'll leave you our invited speaker. Bye, Julia. Take care.
Thank you so much. Merci beaucoup, Adnan.
Yeah, you're welcome. Bye.